myoglobin and hemoglobin. Myoglobin, okay, myoglobin is the same, stores oxygen just like hemoglobin does. It binds to oxygen. Hemoglobin is the oxygen transport protein of higher animals. It binds in our red blood cells and transport oxygen throughout our body. Okay. Myoglobin is the oxygen storage in the skeletal muscles. So hemoglobin basically occurs in the blood. Myoglobin occurs in your skeletal muscles. Okay, that's the difference. They both transport oxygen, they both bind oxygen and get oxygen to where it's needed. Okay, so oxygen is transferred from hemoglobin to myoglobin, okay, in the muscle cells, because myoglobin has a stronger attraction, a stronger affinity for oxygen than hemoglobin does. Okay, so when we transport oxygen throughout the body, through the bloodstream, the body transfers the oxygen to the muscle cells where it's bonded to hemoglobin, and that takes over from there. Okay, and this is your the hemoglobin. Okay, you got your four chains. Okay, you got your heme group with your iron in the center. Okay, and that's what holds your oxygen molecules in place. Okay, so the heme group is the component of the protein hemoglobin and myoglobin. They both have a heme group. Okay, that's what holds oxygen to it. Okay, iron 2+ in the heme group is the oxygen binding site. Okay, so we need iron to be incorporated into the heme group of both myoglobin and hemoglobin to be able to bind oxygen. Each hemoglobin chain contains a heme group, which can hold one molecule of oxygen. So every heme group holds one oxygen molecule. Okay, and that's oxygen. That's the iron in the center, and these are what's called porphyrin rings that are surrounding it here, and that's what a heme group looks like. And every one of those heme groups, for every heme group you have in a red blood cell, it can hold one molecule of oxygen. Okay, so here's your red blood cell. Okay, you have, remember, 2 beta chains, which are identical, and 2 alpha chains. Okay, each chain has a heme group associated with it. You can see each heme group right here. Let's see, because it's not being used. Okay, you have 4 heme groups with iron in the center. So 1 heme group for each chain. So one hemoglobin molecule can hold 4 oxygen molecules. Okay. So the oxygenation of hemoglobin in the lungs is favored by the difference in the oxygen partial pressure in the lungs and the blood. In other words, if you're going to the lungs, we'll draw the diagram here too. So blood goes, you're pumping blood into the lungs. Here's your lungs. Let's right and left lungs here. Okay. So blood's coming to your lungs. The blood that's coming to your lungs has already been used by the body to have the oxygen taken out of it, right? The heart pumps the blood through the rest of your body. Oxygen gets delivered throughout all the tissues of your body, then comes back to the heart. The heart pumps it back to the lungs. The lungs put oxygen back in the blood, goes back to the heart, and then it pumps out the rest of the body, giving you oxygen. So the blood coming to the lungs has a low oxygen content. A lot of it's already been used up. Okay. The air inside your lungs, we breathe the air, has a high oxygen content. So when the blood comes in contact with the capillaries, with the alveoli of the lungs, oxygen is low in the blood, high in the lungs. So oxygen flows from the lungs into the hemoglobin of the blood just by diffusion, just because of the difference between the partial pressure of oxygen. There's more oxygen in the lungs than there is in the blood, so oxygen naturally flows from the lungs into the bloodstream. And that's why oxygen in our blood. Okay. So the partial pressure in the air in the lungs is about 100 millimeters of mercury. The partial pressure of oxygen in the blood is about 40. So if it's in the lungs, you're about 100 millimeters of mercury, and in the blood, you're about 40 after you have deoxygenated blood. So if you're pushing with 100 and you're probably pushing back with 40, the 100 is going to win out every time. So we're going to push oxygen into the bloodstream from our lungs because of the difference in partial pressure of oxygen. Okay. So oxygen diffuses from regions of high partial pressure to regions of low partial pressure. Okay. So it goes from high partial pressure in the lungs to low partial pressure in the blood, and then saturates the hemoglobin. It enters red blood cell and binds to the iron ion of the heme groups of the deoxygenated hemoglobin, forming oxyhemoglobin. So when we have hemoglobin in the red blood cells. When oxygen fuses from the lungs into the blood, into the red blood cells, it's termed oxyhemoglobin, which means oxygen is attached to the hemoglobin now. So that's our term, oxyhemoglobin. Okay. This binding happens to actually bring more oxygen into the blood. Okay. Once it binds to it, it allows for the partial pressure to keep pushing more oxygen into the blood. In other words, the lungs are pushing the oxygen into the blood because the blood's low in oxygen, right? So now the hemoglobin absorbs that oxygen from the blood and puts it in the red blood cell. That keeps the blood low in oxygen. So more oxygen is pushed from the lungs into the blood. Okay? So it actually brings more oxygen into the blood by depleting the oxygen, by moving it into the red blood cells, forming oxyhemoglobin. Okay? Now, similar system has to exist for transfer from mother to fetus. Now, there's got to be some differences here, because if the fetus and the mother had the same affinity for oxygen, the fetus would never get any. Right? So it's got to have a different way of adhering to it, of attracting oxygen to it. Okay, so a fetus receives oxygen from its mother by diffusion across the placenta. Okay, if both the fetus and the mother have the same type of hemoglobin, the process would not be efficient because they both have the same kind of the rate of attraction for it. Because the hemoglobin of the fetus and the mother would have the same affinity for oxygen. That means it wouldn't transmit to the fetus very well. That's not what happens. Okay. The fetus has a unique type of hemoglobin called fetal hemoglobin. Okay. This unique hemoglobin allows a greater affinity for oxygen than does the mother's hemoglobin. So when you get to the placenta now, you got fetal hemoglobin inside the fetus, and you got maternal hemoglobin inside the mother. Okay, the fetal hemoglobin has a greater affinity for oxygen, so it's going to pull that oxygen away from the mother into the fetus. That way, the fetus is going to get the oxygen that it needs to develop. So the big thing is fetal hemoglobin has a greater affinity for oxygen than normal hemoglobin. Oxygen is therefore efficiently transferred by the circulatory system from the lungs of the mother, ultimately from the lungs of the mother to the fetus. Okay. So the biosynthesis of fetal hemoglobin stops shortly after birth. So once the baby is born, okay, it stops the production of fetal hemoglobin. The genes encoding for fetal hemoglobin are switched off, and the genes coding for adult hemoglobin are switched on. So at birth, the baby's going to stop making fetal hemoglobin and start making the normal or the regular adult hemoglobin by turning off the gene for fetal hemoglobin and on the gene for adult hemoglobin. Okay. So why is oxygen efficiently transferred from hemoglobin in the blood to myoglobin in the muscles? That's what we just talked about. Okay. Myoglobin has a greater attraction for oxygen than does hemoglobin. So it allows efficient transfer of oxygen from the bloodstream to the cells of the body. Okay. So myoglobin attracts it more than hemoglobin does. Okay. How is oxygen efficiently transferred from mother to fetus? We just talked about that too. The fetus receives oxygen from its mother by diffusion across the placenta. The fetus has a very unique hemoglobin called fetal hemoglobin. Okay. The fetal hemoglobin has a greater affinity for oxygen than does the mother's. That way, oxygen is more efficiently transported just through the circulatory system from the lungs of the mother into the womb, into the fetus. Hemoglobin variants, when we have run into problems. Okay, a fetus has a unique type of hemoglobin with slightly greater affinity for oxygen than typical hemoglobin, right? That's our fetal hemoglobin we talked about. Okay, another variant is sickle cell hemoglobin, causes red blood cells to appear altered, as in the following scanning electron microscope that you're going to see. Okay, this is a normal red blood cell, round, circular, biconcave. Oxygen can be absorbed in there very easily. This is a sickle cell, like in sickle cell anemia or sickle cell disease or sickle cell trait. That's what a sickle cell looks like. You can see that's not going to absorb oxygen like the other one. And also look at the shape of it. Capillaries are about the same diameter of a red blood cell for maximum absorption. And that's going to just clog up the system. That's not going to flow through very easily. That's got points and edges. It's in the shape of a sickle. Okay? So it's going to block the blood flow, and it's not going to absorb oxygen. Okay? So sickle cell anemia is a human genetic disease. First appeared in the tropical West and Central Africa. Okay, it affects about 0.4% of African Americans. Okay. These individuals produce a mutant hemoglobin known as sickle cell hemoglobin. Okay. Sickle cell anemia receives its name from the sickle appearance of the red blood cell that form when they have sickle cell anemia. When you have the condition, you get that sickle appearance of that red blood cell. The sickle cells are unable to pass through the small capillaries. Again, red blood cells are round and smooth. They can flow right through the capillaries. When you have that sickle shape, you got points, you got edges. They're going to dig in and they're going to block the capillaries. Okay? Circulation is going to be hindered. So they're not going to absorb oxygen correctly, and they're not going to pass through the body through the capillary system correctly. They're going to back it up. Okay? This results in damage to many organs like the bones, the kidney, and can lead to death at an early age. Okay? Because you're not oxygenating the organs that you need to. Okay? Sickle cell hemoglobin differs from normal hemoglobin by a single amino acid. Okay? So one amino acid in that entire protein chain is different. And that changes everything. Okay? So one nucleotide in the DNA switched and it coded for a different amino acid. And it changed the whole thing. Just one single amino acid changed the whole structure because it changed the shape. In the beta chain of sickle cell hemoglobin, a valine, okay, which is hydrophobic amino acid, replaces a glutamic acid, which is a negatively charged amino acid as a base. So you've gone from a polar amino acid to a non-polar or hydrophobic amino acid. So the bond, when it forms to shape, is going to be completely different. It's not going to form those same bonds, and it's not going to make those same shape. Because remember, shape depends upon the primary structure, the order of the R groups. Sickle cell anemia occurs in individuals who have inherited the gene for sickle cell hemoglobin from both parents. It's a recessive gene. If you inherit from only one parent, you're a carrier. If you inherit from both parents, you can have sickle cell anemia, you can have the disease. Afflicted individuals produce 90 to 100% defective beta chains. Okay? Once that gene is switched, that's it. Okay? Individuals who inherit one normal gene and one defective gene produce both normal and altered beta chains. Okay? About 10% of African Americans carry a single copy of the defective gene, condition known as sickle cell trait. You know, they carry it, they don't have any manifestations of it. Their red blood cells still look normal, okay? But they have sickle cell trait. They carry the disease in their gene. Okay? Although not severely afflicted, they have a 50% chance of passing the gene on to each of their children. It's a recessive gene. It's going to make a, in other words, if you have a recessive, you have a dominant gene or recessive gene, one of those will be passed on to children. One's normal and healthy. One's the one that's coded for sickle cell anemia. Right? Okay, why you probably ask yourself, if it's such a bad disease, what has such huge results, why is it still present? Why doesn't evolution filter it out? Because if you have sickle cell trait, you're resistant to malaria. Okay? When you have the trait, that makes you that gives you some resistance to malaria. Okay? So in some parts of Africa, up to 20% of the population has sickle cell trait. That's because sickle cell trait gives you resistance to malaria. Okay? In those same parts of Africa, One of the leading causes of death is malaria. Okay. The presence of sickle cell trait is linked to an increased resistance to malaria because the malarial parasite cannot feed directly on sickled red blood cells. So if you have the trait, it gives you an advantage over everybody else of not getting malaria. So that way it's carried through the population, through evolution. So people who have sickle cell disease die young. Those without sickle cell trait have a high probability of succumbing to malaria. So in those areas of Africa where we have high incidence of malaria, you want the sickle cell trait. So you can still function normally with that. So occupying the middle ground, people with the sickle cell trait do not suffer as much from sickle cell anemia and simultaneously resist the deadly malaria virus. Okay. Because those with sickle cell trait have a greater chance of survival and reproduction, the sickle cell hemoglobin in the gene is maintained in the population. So the people with the trait have a genetic advantage over everybody else because they don't succumb to malaria. So that gene is continued on and populated through the population. All right. Any questions on our red blood cells? Myoglobin, hemoglobin, oxyglobin, all that good stuff. All right, we have proteins in our blood. Blood plasma contains about 60 to 80 grams per liter of proteins, alpha through gamma. The most abundant is albumin, the most abundant protein in the blood. It's a transport molecule for poorly soluble metabolites like bilirubin, calcium, fatty acid. It helps transport that to the cells and into each cell through the cell membrane. That's what albumin's function is. The alpha globulins are the next most abundant, and they include glycoproteins, high and very low density lipoproteins, that's your cholesterol, stereoplasmin, and prothrombin. Okay. Then you have beta globulins, which include transferrin and low density lipoproteins. Okay. Fibrinogen is a protein involved in coagulation of your blood, works along with the platelets in your blood to form a blood clot during times of injury or damage to your blood vessels. Okay. That's one of the proteins that circulate through the blood, is fibrinogen. Finally, the gamma globulins are synthesized by the B lymphocytes. Okay, B lymphocytes are involved in your immune response. The gamma globulins are your antibodies that get produced. Okay, those are the ones that fight viruses, bacteria. Those are the ones that are activated by vaccines for viruses like the COVID vaccine, flu vaccine. What it's doing is activate your globulins, your gamma globulins, so they can respond to an infection in a quicker manner than if they weren't there. Most remaining plasma proteins are synthesized in the liver. A hallmark of liver disease is reduced amounts of one or more of the plasma proteins. That's how we do a CBC, a complete blood count, and you look at the amount of plasma proteins. And if there's a decrease, one of the first things you're going to look at is for liver disease in your patient. Denaturization of proteins. So you have proteins. Proteins perform a function. Okay, to destroy a protein, you can denature it. If you denature a protein, it will not have its shape or its function any longer. Okay. So extreme temperature, extreme pHs have a drastic effects on protein conformation, their shape. Okay. Because your body's maintained at a certain temperature, a certain pH. When you get sick, when you get a fever, okay, what happens to you when you try to eat something? Does it feel good? Does it feel right? No. When your body runs a fever, you're elevating your temperature. You're denaturing the enzymes in your body. They can't break down the food anymore. So when you eat, it just sits there, makes you feel even sicker. So when you're sick and you're in a fever, you don't want to eat. You don't have an appetite because your enzymes are not functioning. They're not going to break down that food. You've got to keep your body within a certain range for enzymes to work, certain pH range and certain temperature range. Okay, so denaturization occurs. Okay, denaturization is the loss of the organized structure of a globular protein. We show you how all those proteins folded, right? You had disulfide bridge, you had hydrogen bonding, you had hydrophobic interactions, you had salt bridges. Those all form this globular protein. This shape makes it work. When you denature it, you heat it up, it breaks that shape apart, you lose that shape. It breaks those hydrogen bonds, those salt bridge bonds, and you lose its shape, and therefore it's not going to work. That's denaturization. Okay? It does not alter the primary structure. Because think about it, all it's doing is breaking the bonds between the R group. It's not modifying the primary structure at all. It's just modifying the quaternary and the tertiary structures. Okay, those are all staying the same. Oh, why is that happening? There we go. Okay, so you have helical proteins. This is their shape. You add heat and they spread out. Okay, again, the R groups are still in the exact same pattern. The primary structure is still the exact same. You haven't broken any polypeptide linkages or bonds there, but you change the shape. So you have denatured the protein. And once they're denatured, they clump into groups. So you don't get them back again. Okay? So denatures them, and then those denatured groups clump into groups, forming just the main clump that doesn't do anything. So you have your regular shaped proteins, you heat them up. You can do that by lowering a rate. The lowering a rate in the pH also can cause this. Denatured proteins become a coagulated protein, non-functional. Okay, so these are non-functional once they achieve this step. Okay, poisoning of proteins. Okay, because structure and 3D shape matter and activity, proteins are very susceptible to changes in conditions, just like with temperature. Okay, hydrolysis. Hydrolysis is breaking down into smaller peptides or breaking down to amino acids. You break the protein chain down. Changes in pH can cause this. Enzymes can cause this. Temperature can cause this. Okay, this allows you to remove water from between that polypeptide linkage and change it into a small peptide or break it down to amino acids. Again, change in pH, temperature, enzymes. Denaturation is a breaking up the 3D shape. So hydrolysis is used like when you eat proteins. Okay, we have to break it down to get the value for it, right? We break it down into smaller peptides or individual amino acids as you break proteins down into, so you can utilize them in the body. Okay, and we get change in pH, enzymes, and temperature all have that function. Denaturization is only break up the three-dimensional shape. It does not make it into smaller chain. It does not release amino acids. The primary structure stays the same with denaturization. Again, temperature, heavy metals can cause it. Heavy metal poisoning, detergents can cause it, and organic solvents can cause it. Okay, and mechanical stress can cause it to break down. Okay? So any of those can denature a protein. Temperature, heavy metals, detergents, organic solvents, mechanical stress. Okay. The effect of temperatures on proteins, as the temperature of a solution of protein increases, okay, the first effect is the increase in the rate of molecular movement of the individual molecules in the solution. So when you heat something up, the molecules in the solution move faster. That's what first happens, which makes sense, right? And putting more kinetic energy into it. Then as the temperature increases further, the bonds within the proteins become to vibrate more violently, and those bonds are going to start to break. Eventually, the weaker hydrogen bonds and hydrophobic interactions, those are the bonds that hold our tertiary and quaternary structure of our proteins together, are disrupted. The characteristic three-dimensional conformation or shape of the protein becomes disorganized. So you have denaturization, and then you got coagulated proteins. Okay, so when you heat it up, it increases the speed of all the molecules in there. Keep going further heating, it starts to shake those bonds, and keep going that eventually breaks those bonds loose, the weaker bonds that are holding the three-dimensional shape of this molecule together, and then it becomes denatured, and then it get coagulated proteins. pH. Okay, if a protein has a net charge of +2, okay, you add a base, some protein and amino acid groups lose their protons, and you end with a net charge of 0. Okay? So you can add a base or add to an acid, change the pH. You can change the charge on a protein by adding a proton to it. If it's an acidic proton, if it's in an acidic solution, you can add a proton to it. If it's in a basic solution, you can lose a proton and change the net charge on your protein. If a protein has a net charge of -2, adding an acid, okay, can give you a net charge of 0 because you're adding protons back to it. If it's a +2 charge, you're adding a base. Okay, you're donating protons back onto it. If it's an acid, negative charge, you're removing protons. Okay? In either case, as you alter the pH, the proteins will unfold due to the change in repulsion and destabilization interaction. The R groups, okay, that were holding this protein together are going to have different charges. If you take it from a +2 charge to a neutral charge, you're not going to have that polar charge anymore. So your protein is going to denature. If you go from a -2 charge to a 0 charge, you're losing a charge again. You're turning a polar group into a non-polar group, and it's going to break or dissolve that bond that was holding that three-dimensional shape together. Okay? In blood or other tissues, denatured proteins can no longer carry out their function. Okay? Denatured, once you change the shape, it's not going to function anymore. Okay? So this is a net charge of +2. You can see the NH3 group here, NH3 group here. Okay? You add OH. OH, negative hydroxide, absorbs, wants to collect a proton to turn it into water. So it takes that proton away from here. All right, where am I going here? No, it takes the proton off that group there and this group here. So now you have NH2. Okay? So now you've lost that net charge. Okay? So now you have one charge of +2, which is polar, to a net charge of 0, which is nonpolar. Same thing on the other one. If you have a net charge of -2, you have a COO minus here, CO minus here, and then we add 2 H pluses. So the H pluses protonate the acid, okay? And then you lose your negative charge here. Okay? What that does is takes a polar R group and converts it into a nonpolar R group. So if there's a bond in there that was holding it together, it's now going to be broken, and that will denature your protein. That's how pH denatures proteins. Okay. When the blood pH drops too low, becomes too much acidic, blood protons become polycations. There's extra positive charges around. Okay. Similarly, when the pH rises, okay, it gets less acidic, more basic. The proteins become polyanions, negative charges. Okay. In either case, whether you increase the negative charge or the positive charge, okay, the protons will unfold because the charge repulsion of loss of stabilizing ionic interactions. You're losing that stabilizing factor. Whatever bond you had, whether it's a polar bond, ionic polar bond, or sulfur dibridge bond, disulfur bridge bond. Okay. Whatever you do, if you change that repulsion of it, change those charges, it's not going to hold the same attractive forces anymore. You can denature your protein. Okay. Under extreme conditions, the denatured proteins will no longer be able to carry out their required functions, and they form protein conglomerations. Okay. The blood cells would also die as their critical enzymes were denatured. Okay. So once you lose the enzymes that are making your blood cells, okay, they become denatured. Those are going to die also. Because you're interfering with the process of it, it's not going to continue to work. Okay. The hemoglobin in the red blood cell, which you talked about, would become denatured and would no longer be able to transport oxygen. Okay. All right, so those are the effects. Organic solvents, hydrocarbons. Okay, organic solvents are non-polar. Polar organic solvents such as rubbing alcohol, 2-propanol, denature proteins by disrupting hydrogen bonds within the protein, in addition to forming hydrogen bonds with the solvent. Okay? That's why alcohol is used for disinfecting surfaces. Okay. It denatures the proteins, the bacteria, whatever's on the surface, by disrupting hydrogen bonds within the protein. Okay. The nonpolar regions of these solvents interfere with hydrophobic interactions in the interior of the protein molecule, thereby disrupting the conformation of those shapes. Detergents. We talked about this with soap, with micelles that are being formed. How are cell membranes formed? Detergents have both a hydrophobic and a polar or hydrophilic region. Okay. They can disrupt the membranes that way with bacteria. So when a detergent interacts with proteins, they disrupt the hydrophobic interactions, causing the protein chain to unfold. Heavy metals, same way. Lead poisoning, mercury poisoning, okay, form bonds with negatively charged the side chains. Okay, your R groups. Okay, heavy metals have a positive charge. It's going to bond to the negative charge of negative amino acids in a protein. This interferes with the salt bridge formation between amino acid groups of the protein chain, resulting in the loss of its shape. Okay. Remember, you have the salt bridge that formed by the ionic connections. Okay. If the heavy metals are now going to bind to the negative portion of it. Okay. So if you had the shape of your protein, let's say you had your protein here, and you had Na plus, and protein here, well, we had a minus. Okay. And this plus and minus attract, right? So now if we have heavy metal poisoning, mercury or lead, okay, this +2 charge attracts this negative charge here, and it pulls it and opens up this part, therefore eliminating the shape of our protein, causing denaturization and the protein not to work anymore. Okay. So heavy metals would also bind to the sulfur groups of a protein. Okay. Some amino acids have sulfur groups in them. This may cause a profound change in the three-dimensional shape, okay, accompanied by loss of function. You change the shape, you're going to change the function. Okay. So here's your active enzyme. There's the two SH groups here. Okay. You can see the site where something can attach right here and bond to it, and the enzyme can work. Heavy metal poison, the mercury. Sulfur has free electrons around it, just like with oxygen. Has 8 valence electrons, remember. Those are negatively charged. Mercury is a positive charge in bond between the two sulfurs. And then instead of our active site being here now it switches to this active site. So the same substance is not going to be able to bond on this site as it did on the other one. So you've basically taken the enzyme and made it inactive to what it's being used for, because you've changed the shape of the active site by bonding, by changing the bonds of the two sulfur atoms together inside the site. Because all our heavy metals have positive charges on them. Okay. So lead compounds are quite toxic. Okay. You excrete about 2 milligrams of lead every day. Okay. Our intake from air, food, water, used is less than that. So normally you do not accumulate toxic levels of lead. But if you drink from a ceramic mug that has not been glazed properly, if you put coffee in it, the coffee's acidic, it's gonna etch out the lead that's inside of that ceramic coffee mug, and you're gonna get lead poisoning from that, and that does happen. Okay. If intake exceeds excretion, okay, if you take in more than 2 milligrams of lead every day, it's gonna build up in your body, and it will cause chronic, irreversible lead poisoning. Okay. Lead poisoning is a major problem with children in those areas that contain old, rundown buildings because of the paint. The paints we used to use were lead-based. So children eat the lead-based paints, they can get lead poisoning because it increases more than they can excrete from their bodies. Okay. Some children develop a craving that causes them to use unusual things. There's several diseases that do that. Okay. Children with this syndrome called pica eat chips of peeling lead-based paints. Okay. These children can also pick up lead compounds from the streets where they've been deposited by automobile exhaust and other sources. Okay, such poisoning often leads to mental retardation, neurological disorders by damaging the brain and the nervous system. Okay. The U.S. Environmental Protection Agency estimates that lead poisoning contributes to 123,000 cases of hypertension and 680,000 miscarriages annually, and retards the growth of 7,000 children every year. Okay, that's just from lead poisoning. Okay. These problems add about $635 million to the healthcare cost annually. Okay, because of these health concerns, lead salts are no longer used as pigments in paints, and most of the lead has been removed from gasoline. Now we use non-lead gasoline also. Okay, mercury compounds are also quite toxic. In August 1996, Karen Weatherham, professor at Dartmouth College, spilled a tiny amount of dimethylmercury, okay, on the latex gloves she was wearing. The mercury-containing compound permeated the glove and was absorbed by her skin. Okay, a few months later, she became ill and died of mercury poisoning, less than a year following the exposure. So it's nasty stuff. Okay, it can absorb really quickly, and it can alter those enzymes so your body cannot function. Okay, now if you ever had silver fillings in your teeth, silver fillings are silver, zinc, copper, and mercury. Okay, you think now the mercury in the amalgam corporation is not free mercury. It's not mercury dimethylmercury. It's not a poison. It's not a toxin. The mercury in your teeth are not poisoning you. All right. There's a lot of false information out there. There's never been any scientific study to show that that is the case. In fact, if you come in my office and I remove a silver filling and put in a white one, okay, for health reasons, I will lose my license. Okay, you cannot do that. Now, if you come to my office and you want your silver fillings replaced because they look better, go for it. No problem with that. You cannot replace them for health reasons because there is no studies that have ever shown that the mercury is coming apart in those fillings as free mercury. You are not dissolving, you're not losing it. Okay. There's all kinds of fake studies out there, but silver fillings are healthy on your teeth. But when you replace them, I definitely go with the white or the composite fillings. I mean, I haven't done a silver filling in over 25 years now since I've done a silver filling with mercury in it. The other ones are just so much better, so much healthier for the teeth, so much better looking. All right, so Fowler Barley showed that the dimethylmercury can penetrate disposable gloves in 15 seconds or less. Okay. Inorganic mercury salts are less volatile and less readily absorbed by the skin. They're bound to other materials, like in your silver fillings. Okay. They're bound in other compounds containing mercury, but they still present health and environmental hazards. Now, environmental hazards we do have when you bury a body with silver fillings, then that mercury gets broken down, and it can cause environmental problems. So several countries have banned mercury fillings, like Switzerland has, I know. Okay, just because of when you bury the person, that mercury lets out into the environment. That it does do. Hatter's disease, remember Alice in Wonderland, the Mad Hatter? Okay, was a form of chronic mercury poisoning. Okay. There's mercury in the metal bands of the hats that people used to wear, and to be absorbed through the skin, and it would cause mercury poisoning. That's where we got the Hatter's disease from. And that was the Mad Hatter in Alice in Wonderland. Okay. Mercury compounds are used to convert fur to felt for the felt hats. And you would get absorption through the skin of the mercury. Mercury compounds still occur in water systems because large quantities of mercury and mercury salts have been dumped by industries into streams and lakes. So it is definitely an environmental concern. The mercury is taken up by fish, and then we eat the fish. That's why certain fish are not advised for pregnant women to eat, because of the mercury content in certain areas. Okay. Tuna is one of them that have a high concentration of mercury in them where they were feeding. All right, we can also have mechanical stress. We can break them down by force. Okay. Stirring, whipping, shaking can disrupt the weak interactions that maintain protein conformation. Okay. This is the reason that whipping the egg whites produces stiff meringue. Okay, you're taking the egg whites, which is a liquid, and all you're doing is beating it up, right? With a whisk. Okay, and that makes a meringue. Okay, think about it. You're taking these proteins and you're basically knocking them around so much, you're breaking all the weak bonds, you're denaturing the protein, changing the shape, and causing the egg whites to turn into meringue. You didn't know you couldn't make meringue without knowing chemistry, huh? So how does high temperature denature proteins? High temperature does what? Remember we talked about it. It heats up the molecules in the solution. The molecules start moving faster, which causes the bonds to vibrate between the R groups, one to the other. They vibrate enough, they break the protein apart. Protein breaks apart, denatures it, and then they clump together. So high temperatures cause the weak forces when together the tertiary structure of the protein to break apart, causing denaturation. The protein then is non-functional. It depends on its three-dimensional shape to function properly. How does pH cause proteins to coagulate? Same thing. pH changes the charges on one of the R groups. Okay. If the R group has a positive charge, okay, and we take it to 0, we've taken a polar aspect and made it non-polar, or made it hydrophobic. So now that bond that was there is no longer there. It's not going to bond anymore. You're going to change the shape, you change the function, you change the function, it becomes non-functional. Okay. When the pH drops too low means it's going to be more acidic. The H plus increases, the blood proteins become polycations because it has a positive charge now. The protein will unfold because of charge repulsion and loss of stabilizing ionic interactions. Okay. The protein will lose their negative charge and reach their isoelectric point. Okay. And then the charge you're keeping them in solution by forcing them apart will no longer be present. Okay. And the protein is going to start coagulating out of solution then. Okay. Antibodies we talked a little bit about. Those are the gamma globulins. Okay. Those are also part of your proteins that are circulating in your blood. Gamma globulins circulate in your blood and are part of your immune system. Okay. Those are your antibodies. An antibody is also called an immunoglobulin, is a specialized protein that plays a role in your immune response. Okay. Whenever it is invaded by a foreign substance, the body responds by increasing the synthesis of specific antibodies to eliminate the invader. Okay, what happens is bacteria, virus enters your bloodstream. Your body goes, oh, that's not supposed to be here. So the B cells, which is part of your antibodies, they stick to that foreign object, the foreign surface of it. It has little spots on the outside that are foreign to your body. It sticks out and a little flag goes up. Then the T cells in your body see that flag and destroy it. So it's marking this as a foreign object. Now, if the virus attacks so quickly, the body can't keep up with it a lot of times, so you get sick. So the vaccines we have out today, like the flu vaccine, if you've ever taken the flu vaccine, it's impossible to get the flu from the flu vaccine. I know people tell they do it all the time. They get the flu vaccine, they get sick. Yeah, you can get sick. You can have a reaction to anything, but they're not going to get the flu from it, at least not one of the ones that are specific for it. Because what happens is we take an attenuated virus, which means it's dead. So we kill the virus, but it still has the outside coating. We inject it in your body. The antibodies recognize that coating as foreign. So your body makes antibodies to mark it. These antibodies circulate your body and wait for that virus now. So now, when the real virus comes along, later on down the line, it gets in the bloodstream and you've got all these antibodies already made for it. So they attack it instantly and you destroy it before it has a chance to take hold. And that's how vaccines work. Okay. Now, can you get the flu, given a flu vaccine? Sure. The flu vaccine from the CDC, they pick 20 different strands for that year that they think are going to be the big causes. Okay. And they inoculate this for those 20 strands. Now, could you get the 21st strand to get the flu? You sure can. Okay. Could they make a mistake and go, oh, we kind of messed up. We didn't count the one that's wiping everybody out right now. They can do that. Okay. But those 20 strands that you were inoculated against, you're not going to get. Your body's going to respond to those. Okay. But that's basically how our vaccines work with antibodies. It triggers the synthesis of antibodies. So the invader is called an antigen, and the antibodies attack the antigens. Okay. Each antigen stimulates the production of a unique set of antibodies. So you have antibodies specific for those 20 diseases, those 20 flu vaccines, those 20 viruses that the flu vaccine is going to protect you against. They're very specific. Okay. Antibodies produced by the B cells, a specific type of white blood cell, leukocyte. Okay. Each B cell makes a specific antibody. And like I said, once it marks it, it tags it, and the T cells attack it and destroy it. Each antibody contains 4 polypeptide chains, 2 heavy chains, 2 light chains, and they're linked by disulfide bonds. That's what forms an antibody. So each chain has a constant and then a variable region that allows you to change for the different antigens it's exposed to. Okay. The constant region of each antibody of a particular class, and it's 5 different classes of antibodies. Okay. They have the same amino acid sequence that forms the basic structure that's going to hold the variable parts together on it. In other words, if this is your antibody here that comes down here, this is all the exact same for all antibodies. And then this upper part here changes to modify the shape to match the antigen that's specifically attaching to. So this is all the same. And then these are your regions that are able to be modified. Okay? Different amino acid systems for different specific antibodies. So each antibody has 2 regions where combined to a specific antigen. This is what they look like: IgG, immunoglobulin, G, immunoglobulin E, D, M, and A. Okay, 5 different types. And you see, this is all the same. And this is the different parts right here. These are your different parts. Okay, that allow you to bind to the different antigens. Okay. So the different types of antibodies and where they're located in the body, you can see IgG. Okay, this is the most common type. Almost looks like Optimus Prime, doesn't it? Those of you into Transformers. IgM, produced first by the antigen invasion, increases transistivity. IgA, in mucosal tissue. Okay, they form dimers, means they form 2 at a time, 2 a pair. IgD is surface receptors for mature B cells. And then IgE is what's involved in your allergic responses. So they all have different responses of how they act in the body. And that's what the cell looks like. Okay. This is a virus cell. In fact, this might be the coronavirus. It sure looks like it. These things on the surface here are the antigens. Okay. Those are what the body responds to. This is an antibody right here. And it's found something it doesn't recognize. It's the same with your body. It found something foreign. So it sticks to it. Now, this is a flag. This is a marker. When a T cell comes along and sees this, that's an indication to destroy the cell. Okay, T cells go through the bloodstream. They look for these little flags that pop up. And once these flags pop up, it says, time to destroy it. And then it marks it for destruction. And the T cells release lysosomes, release enzymatic enzymes, which break down the virus. Okay. Immunoglobulin G, IgG, is a major serum immunoglobulin. That's one that's most common in your serum, in the bloodstream. Okay. Some immunoglobulin G molecules can cross the cell membranes and can pass between mother and fetus. That way you can give the fetus some immunization before birth. This is important because the immune system of fetus is immature. Okay, and they cannot provide adequate protection from disease. That's what happens. If you're sick, do you go see your friend has a new baby? You should say no, because the baby has an immature immune system. It cannot make its own antibodies. It's relying on the antibodies of the mother to protect it while its immune system develops and starts to become functional. Fortunately, IgG acquired from the mother protects a fetus against most bacterial and virus infections that it might encounter before birth, because it's get into the bloodstream. Okay. There are 4 additional types of antibody molecules that will vary their protein composition. We talked there's the 5 different types of antibodies. They all have that general Y shape where they share a common amino acid backbone, and then this modifies the shape of where it can bind to it and flag a pathogen. Okay. One of these is IgM, immunoglobulin M, found in muscle cells. It's the first antibody produced in response to an infection. Okay. Secondary, the B cells produce IgG molecules, which has the same antigen binding region, but a different protein composition in the rest of the molecule. Okay. So they allow you to attack different areas. IgA is immune responsible for protecting the body surfaces of the skin. Okay, the mucous membranes of the nose. Okay, the mouth, oral cavity, your intestines. Okay, so it protects your mucous membranes of the gut, oral cavity, and the genital urinary tract. Okay. IgA is also found in mother's milk, protecting the newborn against diseases during the first few weeks of life. That's why breastfeeding is important, because it transfers not only milk and nutrition, but also transfers immunity, transfers the IgA antibodies that the baby can't produce yet. Okay. IgD is found in very small amounts. It's thought to be involved in the regulation of antibody synthesis and making more antibodies. The last type is IgE, immunoglobulin E. Okay, a lot of times structure was unknown, but it's found in large quantities in the blood of people suffering from allergies. Okay, so they figure this is an overblown immunological response to dust particles or pollen granules. It's how the body responds to it. And when it gets overgrown, it gives you that allergic response. Whereas it doesn't, some people, it doesn't others. Antibodies bind to antigenic determinant sites of the surface of the bacterium. That's what we were saying. On the surface of bacteria, there's markers. They're monosaccharides, they're sugars. Okay, and then different shapes. And we find that shape as it's not recognized by our B cells. So our B cells mark it, they attack it and bind to it, mark it as foreign with the antibody. Then the T cell comes along and destroys the bacteria. Because each antibody has 2 binding sites, the interaction of many antigens and antibodies creates a large antigen-antibody complex that can precipitate out a solution. Okay. This large complex can inactivate the antigen in several ways. Okay. It may simply prevent the antigen from binding to its target, or if it's large enough, it will precipitate out and be degraded by phagocytes like T cells. Okay, so we can dissolve it. The immune system has 3 important characteristics you have to have. Okay, so your immune system, where's my mark? Okay, has to be highly specific. Why does it have to be highly specific? You can't just go through your body identifying things as foreign, because then it's going to destroy your own cells. It's got to be able to tell your cells, and then when something foreign comes along, it has to be able to tell it a high enough specificity to tell the body that that is foreign, that needs to be destroyed. So it has to be very highly specific. You cannot just attack any cell at once. You have to attack only cells that is foreign to your body. Okay. The immune response to each infection to specific or directed against only one disease organism or similar related organism. Remember, the CDC gives you 20 different markers in their flu vaccine. So you're inoculated against those 20, but not against the 21st or 22nd one. It's very specific. It's against that one disease. It has memory. That's the whole key. If we did these vaccines and we'd remember it, it would be useless doing the vaccine. So the antibody has to have memory. It has to go, once it's formed, it has to go, okay, this is formed. I know what to look for now. We've been exposed to this thing. This is a nasty thing for the body. We don't want it. So when it comes into the body with the virus, the body goes bam, and it finds it. So our antibody's already made, response already occurs. So the response has to have memory, or be no sense in taking a vaccine. Once the immune system responds to infection, the body's protecting against the infection by that same organism. So you're not going to get reinfected by it. It has memory. This is really some suffer the same disease more than once. Okay? Most of the diseases we suffer currently, like the common cold and flu, are different strains of the same virus. The virus mutates, and it's a different strain. And if it's a different strain, we don't have antibodies already made up for it. Okay? Each of these strains is going to be new to the immune system. It's going to be a new presentation every time. Okay? It can recognize self from non-self. That's the other important part. You have to be able to tell what is your body and what is not your body. Okay? You have to say what is self and what is non-self. When the antigen or the virus gets in there, your body has to say this is non-self. It's not your own tissue. So when we are born, our immune system is already aware of all the antigens in our bodies. When you're born, your body's already been exposed to your immune system. So it's already marked, it's already found that stuff and said, okay, all this stuff I'm seeing now, this stuff is not foreign to me. So we're not going to develop antibodies against it. Okay. Now, there's some very specific exceptions. Your nervous system is never exposed to your bloodstream during development. So if your nervous system becomes exposed to your immune system will destroy it. That's an autoimmune disease. Okay, it's finding tissues that weren't, it is your own tissue, but it never was exposed to it during development. So you don't know it's your own tissue. And you break down your own body tissue. So these recognize as self and do not attack it. That's the key point. Every antigen that is not classified as self will be attacked by the immune system when it's encountered. Okay? So when you're developing, your body goes through, these antibodies go through your whole body, and it makes a list. It checks all your tissues. It goes, okay, here's my list. When I see these, I don't make antibodies. These are for me. These are me. When I see something different, we make antibodies. Okay. Some individuals suffer from a defective immune system response that allows it to attack the cells of its own body. That's an autoimmune disease. Okay. The result is what's called an autoimmune reaction, and autoimmune reactions can be fatal because you're destroying the body. Okay. An important part of the immune response is the lymphatic system. Okay. This system can return components of the interstitial fluid to the bloodstream, composed of veins and capillaries, but no arteries. So there's a bunch of tubes runs through your bodies along with your veins and arteries, okay? But it's created and it drains that fluid back into your venous system, back into your body. Okay? And it looks for and analyzes threats in the body in the same way as in the bloodstream. Interstitial fluid is absorbed into the lymph capillaries, which is called lymph. Okay? Lymph is a tissue in our capillaries. Now, if the lymph tissue becomes blocked, you get diseases like elephantitis, or your leg becomes 7-8 times its normal size. That's because you blocked the lymph fluid, and now the lymph can't go anywhere, and you're producing more lymph, and it swells up where the blockage is. Okay, so your interstitial fluid is absorbed into the lymph capillaries and makes lymph. Then the lymph has to drain back into the venous system. If it's blocked, it will cause swelling, and it can cause tremendous swelling. Okay, this lymph flows into larger and larger lymph veins. Eventually, the two lymph veins empty into the veins of the blood circulatory system. So it drains back into your venous system. But it's full of all kinds of things looking for infection. Lymph nodes act as filters of the factory for production of some forms of leukocytes. Those are white blood cells that fight infection. If you're ever sick, if you put your hands under your mandible, under your chin right here, you feel those glands that are swollen, right? These are your lymph glands. They're swollen up because your body's found an infection, and these things are making antibodies like crazy. They're becoming larger, making more antibodies to try to fight the infection. That's how they get swollen up. They become larger because they're starting to work now and make a whole bunch more antibodies to fight this infection that you have. Those are your lymph nodes. The leukocytes located there remove the dead cells, bacteria, and other foreign cells from the lymph. So they dissolve it and get rid of it. Okay, lymph nodes are the site of antibody synthesis. That's where you make antibodies. Like I said, when you get sick, we expose that virus, these lymph nodes kick into overdrive and they become larger and swollen. And that's what you can feel. You can feel like that right under your chin. Those are the cervical lymph nodes. You also have them under your arm, your groin area, around your stomach. those are high concentration of these lymph nodes. Okay, but the most common ones you're going to find are right underneath your mandible. Okay, that you're going to feel when you get sick. Lymph nodes are lumpy enlargements in the lymph veins because they're making more antibodies, IgE. Okay. Sometimes the nodes are so infected that filtering out bacteria from an infected area, they become swollen. And that's what we feel. Someone suffering from a sore throat may also exhibit a swollen or tender lymph nodes in the neck because it's producing those antibodies to fight that infection. Is it possible to prime the immune response? Yes, and that's what we've been talking about with a vaccine. Vaccines contain a weakened form of the antigen, dead bacteria. It's attenuated, it's dead. Will cause him to build a certain level of antibodies for this particular antigen. So like I said, the CDC picks 20 different antigens, and that's our shot. And if you get through those 20, already have antibodies in our system because of this to fight that infection. Okay, and the same antigen is then subject to attack by more virulent form of the antigen. The body responds more rapidly and effectively to its presence. So it responds quicker, and it doesn't get the virus bacteria a chance to get a hold on you. Okay. So having practice on the dead invader, the system can easily handle a live one. It's gained an immunity of that particular organ because it's made those antibodies and it has memory. It has those antibodies already made and waiting for it. Unfortunately, the body can't tell good antigens from bad ones all the time. Okay, it doesn't always work like that. We want it to respond to a viral infection, but we wish it wouldn't attack purposely transplanted tissue, like when you get a lung transplant, a heart transplant. Okay, your body's going to think that is foreign. It's going to attack against it. Okay, so we put it on what are those drugs called? The drugs that anti-immune drugs, and they decrease your immune response. Don't blank on the exact name of them right now. The host body will respond to a donor skin graft, a kidney transplant, or a heart transplant, as it does to a virus, by generating antibodies to attack against it, because it's foreign. Okay, the body responds to foreign tissue this way. Process is called rejection, and then you reject the new heart, reject the new lungs, liver, etc. Okay, a major problem associated with organ transplants. Okay, you have to give them certain drugs to knock down their immune system. They don't respond and reject what you just put in them. Okay, immunosuppressant drugs. That's what they're called. I couldn't think of the name of it. Okay, you give them the immunosuppressant drugs. There are drugs that suppress the immune response, but when these are given to a transplant recipient to protect the transplant organ, the patient also becomes more susceptible to infection. Okay, so think about it. You got a transplant organ, and now you're going to give them drugs to slow your immune system down. You're going to make yourself susceptible to all the other infections out there too. So it's kind of a double-edged sword. You have to be really careful with it. So you've got to stay isolated for a while and keep away from other people, keep trying to limit your exposure to different viruses. Okay, if you turn off the immune response, you turn off both the good and the bad aspects of it. Okay, there's a wide variety of diseases associated with the immune system. An overactive or misguided immune system can attack its own body organs. That's our autoimmune disease we talked about. Okay? When you break down your own tissues. The system senses something as foreign, even though it isn't. Okay? In multiple sclerosis, the body attacks and destroys the myelin sheaths of the nerve cells. While in arthritis, it is connective tissue in the joints that are being destroyed. So multiple sclerosis, it affects the sheath, so your nerve impulses don't travel as quickly as they used to. So your coordination becomes extremely hindered, okay? Because your nerve impulses don't travel quick enough to get to the brain. Arthritis destroys the connective tissue in the joints that allows you to bend and move your fingers without pain or discomfort. It keeps the bone from grinding on each other. Okay. Type 1 diabetes. Insulin-producing cells of the pancreas are the target. Okay, your body destroys the insulin-producing cells, so your body cannot produce insulin in response to food. Opposite end of the scale is acquired immunodeficiency syndrome, AIDS. Okay. In this deadly affliction, the immune system is destroyed by the HIV virus. Okay. The victim then, whoa, how do I do that? Let's hit that 240 times. Okay, so the victim then succumbs to a variety of infections or to a rare form of cancer, like Kaposi sarcoma. It's really common with AIDS patients. If you're doing dentistry, you'll see these purple little lesions in the mouth. They're called Kaposi sarcoma. Almost always with just you see it in AIDS patients because your immune system is shut down. They get these different types of infections that are rare or you don't usually see. Dietary protein and protein digestion. Okay, we talked about proteins. We talked about their primary structure, secondary structure, tertiary structure, core structure, how they function with enzymes, how they function as regulatory proteins, how they function as structural proteins, how we can denature them, how we can break them apart. But you got to remember, when people talk about diet, we talk about protein, and that's exactly what we're talking about. This is our dietary protein, well, how much we have to bring in to keep our body functioning and what it does, how we break it down. Okay, we've got to be able to break down these proteins to provide fuel for our bodies. So proteins have several dietary purposes. Okay? We oxidize proteins to provide energy, to form ATP. Amino acids liberated by hydrolysis can be used in biosynthesis. Remember, a protein is only a long polypeptide chain made of amino acids joined by peptide linkages or peptide bonds. Okay? We hydrolyze those bonds, we add water back to it. We put an H on one or H on the other, we open that back up, and we break our amino acids back into individual amino acids. And we can use those amino acids to make proteins for our body. Okay? Amino acids which cannot be made in the body are essential amino acids. Real important. There's certain amino acids our body cannot produce. Those we have to get in our diet. Okay? That's the only way to get them. So certain ones we cannot make, those are called essential amino acids. The ones we have to get in our diet. These are some essential amino acids over here: isoleucine, leucine, lysine, okay, valine, tryptophan. These are essential. These we have to take in our diets. The other ones are non-essential. Remember, there's 20 amino acids total. The other ones are non-essential, which means our body can produce those if we need them to. Okay, so proteins, carbohydrates, fats, they're all energy sources in our diets. Some more efficient than others. Proteins can be oxidized to provide energy. Remember, oxidation is the adding of oxygen or the decreasing of hydrogen. In addition, amino acids delivered by the hydrolysis of proteins are used directly in biosynthesis. So the amino acids we break down the proteins into, we use those amino acids to rebuild our own proteins in the order that we need them. Amino acids are also used in biosynthesis of a large number of important molecules, called the nitrogen compounds. Okay? These nitrogen compounds include our hormones, the heme groups of hemoglobin and myoglobin. Okay? And the nitrogen-containing bases found in DNA and RNA, the pyridines and pyrimidines, the bases that we use that bond to our sugars in DNA and RNA. Okay? We need those nitrogen compounds. And we get those through amino acids. Digestion of dietary protein begins in the stomach. Okay. Each enzyme, the stomach enzyme pepsin, okay, begins digestion by hydrolyzing some of the peptide bonds of the protein. So peptide doesn't break in individual units, but it breaks in small. So say you have this polypeptide chain. Those are a bunch of amino acids. So you break it here, break it here, break it here, break it here. Now we've got that chain. So now we can attack these chains a little bit better because they're smaller and we can break them into individual amino acids. So pepsin is just like a chopping block. It's just non-discriminately chops it in different places. And then specific enzymes used to break down each specific peptide linkage and produce amino acids. So this breaks down the protein into smaller peptides. That's what pepsin does, produces smaller peptides. Production of pepsin and other proteolytic just means digestive enzymes in the stomach. Okay, must be carefully controlled because the enzyme activity would digest and destroy the cells that produce them. So in other words, you have to control the cells making the pepsin and the other digestive enzymes in the stomach. You need to control that because because they're going to start dissolving everything, right? They break down non-discriminately proteins. Your stomach wall is protein. It's going to start dissolving that. So there's control over each one of those to make sure we're not digesting our own stomach and using those cells and destroying them. So it's under exacting control. The stomach lining cells that make pepsin actually synthesize and secrete an inactive form of pepsin called pepsinogen. So we make pepsinogen from those cells. So they're not producing pepsin directly on the cells, so it doesn't break them down. It becomes activated later on. So the cells produce pepsinogen in the small intestine. Pepsinogen has additional 42 amino acids in its primary structure. So you got to take those out to make that enzyme active. So it folds in the correct three-dimensional pattern. These are removed in the stomach to produce pepsin. So pepsinogen produces pepsin. That we're not destroying the cells that are producing it. Okay. Protein digestion continues in the small intestine where the enzyme trypsin, chymotrypsin, elastase, and others catalyze the hydrolysis of peptide bonds at different sites in the protein. We're adding water back into the polypeptide. So we're breaking them at the peptide bond or peptide linkage. And we're breaking them apart. We're using those other enzymes now. And those are more specific enzymes. Chymotrypsin cleaves peptide bonds on the carbonyl side of the aromatic amino acid, and trypsin cleaves peptides on the carbonyl side of basic amino acids. Remember we talked about different amino acids. Some are aromatic, they have a benzene ring. Some are basic, they have a negative charge. Some are acidic, some are basic, they have a positive charge. Some are acidic, they have a negative charge. Okay? And it cleaves it, and they have a carbonyl side. Remember the carbon double bond, carbon double bond oxygen, and then the nitrogen side, the C terminal and the N terminal. So the enzyme chymotrypsin trypsin cleaves the peptide bonds on the carbonyl side of both of those. Chymotrypsin cleaves the carbonyl side of the aromatic amino acids. Trypsin cleaves on the carbonyl side of the basic amino acids. Because it gets very specific and breaks them down into individual amino acids. Together, these proteolytic enzymes degrade large dietary proteins into amino acids that can be absorbed by cells in the small intestine. So you chop it up into smaller chains first with pepsin, then the other one's chymotrypsin, trypsin, etc. Break it down into individual amino acids at specific sites. So amino acids can be divided into two major nutritional classes. Okay? Essential amino acids. Remember, essential amino acids are those that cannot be synthesized in your body. Okay? You have to get them in your diet to make the required proteins for them that they're needed in. Non-essential amino acids. Okay, those amino acids can be synthesized by the body and do not need to be included in your diet. So there's two major nutritional classes of amino acids: essential amino acids and non-essential. Essential ones means you have to get them in your diet because we cannot make them in the body. Proteins are also classified as complete or incomplete. Okay, so proteins derived from animal sources is generally complete protein. It provides all the essential and non-essential amino acids and approximately the correct amounts for biosynthesis. So our animal sources we're real similar to them. They provide the same amino acids that we require. Okay. Proteins derived from vegetable sources like plants is generally what's called an incomplete protein. It lacks a sufficient amount of one or more essential amino acids. Okay, so the animal proteins, okay, already have the essential amino acids in them. Okay, they have to derive the same things. Plant or vegetable source proteins usually are incomplete. They lack a sufficient amount of either one or more of the essential amino acids. So people who want to maintain a strictly vegetarian diet, for whom animal protein is often not available, have the problem that not a single high protein vegetable has all the essential amino acids to ensure sufficient daily uptake. So that doesn't mean you can't eat healthy as a vegetarian. What it means is it takes more effort to do it right, because you have to make sure you're getting those essential amino acids from the different sources. You're not just get it from one source like we do with animal proteins. So the major protein of beans contains abundant lysine and tryptophan, but very little methionine, whereas corn contains considerable methionine, but very little tryptophan and lysine. So you got to do your homework if you want to be a vegetarian, to do it right, to make sure you're healthy, okay? Because you've got to have those essential amino acids. So you've got to make sure to pick in the right amounts each of those types of foods daily to make sure you're getting your essential amino acids. So a mixture of corns and beans satisfies both requirements. So this combination called succotash was a staple of the diet of Native Americans for centuries. Eating a few vegetarian meals each week can provide all the required amino acids and simultaneously help reduce the amount of saturated fats in your diet. Remember, saturated fats are the bad ones. Saturated fats are animal fats. They're the ones with no double bonds, long chain hydrocarbons. Okay, so they stick together. Nothing disrupts their pattern. So they have the London dispersion force that hold them together. And they form clogs in your arteries, or they form a solid, basically. Those are your saturated fats. Why must vegetable sources of protein be mixed to provide adequate diet? Like we said, you got to provide them with the correct amount of essential amino acids. So, like we said before, protein derived from vegetable sources is generally what's called an incomplete protein because it lacks a sufficient amount in either one or more of the essential amino acids. Essential amino acids, we said, were amino acids that you cannot synthesize in your own body. You have to have them in your diet. People want to maintain a strictly vegetarian diet. Okay, with no single, you have to combine vegetables to make sure you're getting all the essential amino acids that you need to ensure sufficient daily intake. What are the common sources of dietary protein? Animal meat is a complete protein. Proteins from plants, beans, and corn are incomplete proteins.